Today we will cover about the cell signaling. Especially we will know in this lecture about GPCR. Okay, which type of signalings are happening in the GPCR receptor and what is the process and where the GPCR receptors are doing the signaling processes. We will cover each and everything about this. From the basic to the advanced, we will cover each and everything. Even you don't know anything, you will understand each and everything in this video. Okay. First, we need to know what is cell signaling. See, the cells need to communicate with each other. As you and me, we both are communicating so that we can understand each other, what we are wanted, what we are expecting from each other. So the same way, cell will try to communicate with each other. In this process, they will release something. Okay, that released thing is known as ligand. Okay, the released thing is called as a ligand, and whoever receiving is known as a receptor. Okay, for example, I am as I teaching you. Okay, so the sender is receiving ligand and the receiver is known as a receptor. Cell having the receptor on the plasma membrane and cell is releasing the ligand in the form of the anything. It could be chemical molecule. Okay, either it could be protein molecule, anything it could be. So ligand is nothing but the releasing molecule and the receptor is nothing but receiving the message. Okay, so why they are communicating? Because as we know, the cell need to divide, cell need to do some sort of things like vision. If you want to see something, then also cell signaling should need to happen. If the cyclin, CDK, any factor, if you want to form any factor, anything, this all requires signaling, then only that all process gonna happen. So we are having 4 types of signaling process based on different way. Here you can see the first one, endocrine, where the hormones are released from the glands. I hope you know, but even though you don't know, then also you can understand. See, if this is our brain, and in the brain we know the pituitary gland is present. The pituitary gland will release directly hormones inside the bloodstream. Okay, this is our bloodstream. Okay, so what will this hormone will do? It will travel through the bloodstream and go towards the targeted cell and attach on the receptor and do the signaling process. So pituitary gland is commanding some another cell to do these type of thing in your cell. So this is what happened with the help of hormones in the endocrine. So the traveling process from the pituitary to the another cell is known as endocrine. But when we are talking about the paracrine system, there are neurotransmitters are traveling. Neurotransmitters are nothing, not neurotransmitters traveling, they are the neurotransmitters are present. For example, in our head region, the brain cells are called as the neurons. Okay, they are in this structure. So these neurons will secrete some sort of the neurotransmitters which will travel to the another neuron and attach, and signaling will happen in the cell. So this is known as paracrine. What is the difference between endo and paracrine? See, here in the endocrine, the traveling process is in the bloodstream. It could be from the pituitary gland to the adrenal gland. So the traveling is too long. But in the paracrine, we can see the nearby cells, like nearby neurons are giving signaling and receiving. So that's why we are calling it as a paracrine. And we are having another signaling process known as autocrine. In the autocrine, we can say the cell is having the receptor on the plasma membrane. Also, this cell only will secrete some sort of the messengers, okay, ligands, which will attach on the receptor, and then the receiver also this cell only. So we can call it as a self motivation, self lovable. These type of things is happening in the autocrine. We can take example like T cells here in this autocrine condition. So autocrine condition is nothing but one cell is receiving the ligand, and that cell only responsible for the receiving the message also. Okay, sender and receiver both are happening in one single cell known as autocrine. And in the juxtacrine signaling, we can say there are two cells. Okay, they both are get attached and the ligand and receptor into each other. So whenever the needed time, then only the signaling will happen, which we can see in the notch signaling. Okay, this is the difference between the different types of the signaling. Endocrine, where the hormones are releasing; paracrine, neurotransmitters are releasing, and they are traveling in a little distance. Autocrine means self-sufficient; they are releasing for themselves only. Juxtacrine, the receptor and ligand will completely into each other, and whenever the needed time, they will do the signaling. It comes under the notch signaling. So till we now understand. Here what is the ligand? There are two types of ligands are present. Okay, so this is our cell, this is our second cell. Okay, A cell and B cell. A cell is known as hydrophilic, B1 is lipophilic. Okay, you can see based on the name you can understand hydro is nothing but water and lipo is nothing but lipids. Philic means nothing but the lovable. Okay, so hydrophilic means it is lovable to the water and lipo means lipid lovable. Okay, so these are nothing but loving to the water, it can interact with the water, you can say. So the example of hydrophilic condition is insulin and adrenaline. So we can say the adrenaline and insulin, they both can interact with the water easily, but they cannot interact with the lipids. So the lipid lovable like steroid, estrogen, progesterone, these all are comes under the lipophilic. Here when we try to understand what really happening in this scenario, when you try to know about the hydrophilic condition, we know very well outside the cell the bloodstream is present. Okay, here the blood is present, and in blood the huge amount of H2O water molecules are present. So in the hydrophilic condition, these molecules, the receptors are present on the surface of the plasma membrane only. So the ligand of the hydrophilic ligand will come and attach on the plasma membrane itself. Okay, they will not enter inside the cell, known as a hydrophilic condition. But when we are talking about the lipophilic condition, in the lipophilic condition, what really happening here, you can see the receptor is present inside the cell and ligand will come and attach on in the cell only. Okay, so here the ligand is able to travel through the plasma membrane. Why they are traveling through the plasma membrane? Why they are not traveling in the plasma membrane? Because see, the H2O molecule cannot interact with the lipid, because the lipid is present in the plasma membrane, we know very well. So our plasma membrane is made up of lipids. So the H2O water will not interact with the lipid, so it will remain in the water region. But our lipid molecule can interact with the lipids, so it can easily travel through the plasma membrane and interact with the inside receptor with it. So this is what happening. Also we can say why this is not present on the plasma membrane, means lipophilic is not present on the plasma membrane, because we know very well here on the plasma membrane, if it will present on the plasma membrane, our lipophilic hormones should need to, if a receptor is present on the plasma membrane, so what will happen, our receptor should need to present on the plasma membrane and ligand should need to bind on the plasma membrane itself. But here the blood is having lots of H2O, so lipid can't be interact with the water, so that's why here the plasma membrane is not having receptor. In this lipophilic condition, the receptor and lipophilic ligands both are present inside the cell. Okay, in general I can say in the both condition we can say there are two cells, one cell have receptor on the plasma membrane, another one is receptor inside the plasma membrane, and this can be outside because the water is present outside, so water molecule can interact. This cannot travel inside the cell because the plasma membrane is made up of lipids, so it can't interact with the lipids. But here in this condition, the water is present outside, so the lipid molecule will not able to attach with it, so that's why it will able to travel. Here we can say it has the ability to travel through the plasma membrane because this is ligand also made up of lipid, and also the plasma membrane also made up of lipids. That's it. Okay, I hope you got it. Let's go here in the another thing. For example, my cell is only attaching on the plasma membrane side. It will say I don't want to go inside the cell. So what will happen? This should need to give signaling inside. Who will give? So here the secondary messenger will come in this scenario. Okay, in this scenario, secondary messenger will come and give the second to the another particular protein, so the signaling gonna happen inside the cell. Okay, you can think like this: if this is your home and your friend is not able to come inside because of some reason, and you want to know someone is coming, going, one of your friend will come, but when, how, you don't know. You must need to know the message that your friend is outside or not. So for that, either you need to keep a camera outside, or you need to say to someone who's staying outside, so they will inform you come and give the information, right? So same like that, what will happen, the receptor is binding, but who will give the message to inside the cell, so that signaling will happen. So here the secondary messenger will present in the cell, so that it will give the message to the inside the cell. This is what happens for our secondary message in the cell cycle or anything. There are some camp, cgmp, ip3, dag and calciums are present. They all are responsible as a working as a secondary messenger. All together is not working. Some scenario where 2 molecules or 3 molecules are working as a secondary messenger and giving them signaling inside the cell. So what really happening? So any one of them or any 2 of them or any 3 of them can give the message. And we also must need to know our receptor related things. So think this is our plasma membrane. On this plasma membrane we are having a receptor like this. Okay, if there are some proteins like alpha, one single protein is present, we call it as a monomeric condition. So the receptor is get attached with some proteins. If only single protein is present, we are calling it as a monomeric condition. But if here in this condition with the receptor we are having 3 subunits are present, 3 proteins are present like alpha, beta and gamma, so we are calling it as a trimeric condition. Okay, with the receptor we are having the protein that is known as a G protein. Okay, now we will cover about the protein related thing. The proteins for the signaling is really important. In the signaling process, what will happen, the signaling only can be done with the help of protein. In this condition, what we are learning about, we are activating, we are deactivating and inactivating the protein, so the signaling process going to happen in this all GPCR receptor, or it could be any signaling. Here you can see we are having a protein. You can see here, to activate or inactivate we need to take gap or gift. We are calling this all process as a G protein. Okay, based on the G protein, what we are doing here, on the protein if GDP is attached, we are calling it as an inactive protein, and if GTP is attached, we are calling it as an active protein. Okay, in this condition what really happening, we are having 2 things. Okay, one is gap, which is responsible for the removing of phosphate from the protein. Here you can see the protein is present. On this protein GTP is present and it is in the active form, but these gaps will remove the phosphate, so the phosphate gonna remove. So what will happen, the phosphate is removed means it will get converted into the GDP, so the protein will be get inactive in the condition. But again I want to activate for the signaling process. GEF will come and attach the phosphate on the protein, so the protein will again get the GTP and it will get activation process. So the inactivation is done by gap and activation will done by the GEF. Okay, phosphate, it is giving the phosphate. So this is one way to activate, and another way we are having protein kinase. As you know very well, the kinase means nothing but the phosphorylation. If you know the cell cycle, you have already watched, then you must know kinase means nothing but phosphorylation process. Here what we are doing, we are doing the phosphorylation with the help of ATP, means energy molecule. So one protein where this is in the inactive condition, but we are attaching the phosphate on it, so the phosphorylation process is happening here on the protein, known as a protein kinase activation. Another two conditions like protein phosphorylase and protein phosphatase conditions are present. You can see this protein is getting phosphate from the outside, so it will be get activated. Okay, but here in the phosphatase condition, the already activated phosphorylated protein is get removed phosphate and the inactivation of the protein is happening. Okay, so we can say the phosphorylase is adding the phosphate on the protein and phosphatase is removing the phosphate from the protein. Okay, this is what happening in this condition. And last one is protein phosphodiesterase, where we are breaking some diester bonds on the protein, so the activation inactivation process going to happen here in this condition. Okay, and you must need to know, for example, this is our protein. Okay, this is a protein, and in some condition what will happen, the phosphate present means activation. Okay, and in some condition same protein but there is no phosphate is also called as activation. Okay, it would be critical to understand, in some condition attachment of phosphate means inactivation. This is our protein and inactivation process. If in some proteins the not presence of the phosphate could be inactivation. Okay, so this is what happened based on the different scenario situation and signaling processes. The phosphorylation could be inactivation or activation, we can't say. Okay, and we must need to know this also. We have covered about the proteins, also we know about the ligands, but we need to know about the receptor also. This is our plasma membrane. Till here, this is our plasma membrane, and on the plasma membrane the GPCR receptor is present. This is nothing but one receptor which is present on the plasma membrane in this form. Okay, as you can see, it is crossing the plasma membrane. How many times it is crossing? You can count here: 1, 2, 3, 4, 5, 6 and 7. So 7 times it is crossing the plasma membrane. GPCR receptor is crossing the plasma membrane around 7 times. Also we must need to know it is made up of completely protein. So till here today the whole GPCR receptor is made up of protein. When we call the protein it is made up of protein, it means it have N and C terminal regions. Okay, with that there are some loops are present. You can see here these are nothing but loops. Okay, these loops are called as E1, E2, E3 and E4. Whenever ligand will come, it will attach in between the E3 and E4 loop. Okay, after attachment of the ligand, the C terminal get activated. Also here the 3 subunits present, as we have already know, alpha, beta and gamma. 3 subunits are in the trimer condition. Okay, trimer condition, they are in the trimer condition, they will be present beside the GPCR receptor. This is what about the GPCR receptor. GPCR receptor is nothing but G protein coupled receptor, we can call it as. Okay, this is all about the receptor, how receptor will look when we are talking about specific about the GPCR only. So let's see what is the signaling process is happening in the cell. So see, the GPCR signaling, we are we are going in the understanding about the GPCR signaling. Here you can see this is our plasma membrane, cell plasma membrane, where the ligand will come and attach on the E and E3 and E4 region, okay, loops. You can see the N and C terminal, two regions are present, and this whole GPCR receptor is crossing around 7 times. Also, it is made up of protein. You know very well each and everything. Okay, but still I am revising you so that you can remember for a long time. And you also must need to know why we are doing here in the GPCR signaling. It is responsible for a sense. The sense like smell and vision, these all things are happen with the GPCR. So whenever the GPCR ligand will attach on these E3 and E4 loop center, what will happen on the C terminal, GEF gonna be get activated. If you remember, the GEF activation means it has the ability to add the phosphate, means it will form GTP. So GTP will attach on the alpha subunit. You can see here the GTP get attached on the alpha subunit. Beta and gamma will remain silent. But alpha, when the alpha get attached with the GTP, it will move towards the adenylate cyclase. You can call it as a adenylate cyclase or adenylyl cyclase. Okay, adenylate cyclase is nothing but older version name, and adenylyl cyclase is newer version. Here, when the adenylate cyclase will get activated with the help of alpha subunit, because of alpha subunit is get activated with the help of GTP. So adenylate cyclase will form CAMP with the help of ATP. So we know very well the cell have lots of ATP energy molecule. Using ATP, it will start forming CAMP. We know very well CAMP is nothing but our secondary messenger here. CAMP, IP3, calcium, these are CGMP, they all are our secondary messenger. So CAMP will go towards the protein kinase A. When we try to understand protein kinase A, this is whole structure is called as a protein kinase A. Here you can see there are two subunits are present. One is catalytic subunit, which is present in the center form, and another one is regulatory subunit, which is present in the yellowish color. So the two subunits are present here in this condition. Okay, and when the CAMP will form, CAMP will come and attach on the regulatory subunit. When the CAMP get attached on the regulatory subunit, the regulatory subunit and catalytic subunit will be get separated. So our catalytic subunit gets separated and regulatory will get separated here. These pKa catalytic subunit go inside the nucleus and it will activate CRIB. With what it will attach, means catalytic subunit will give phosphate to the CRIB. Okay, so that CRIB will get activated. Who is CRIB? Here the CRIB is nothing but CAMP response element binding protein. When we try to understand CAMP response element binding protein, you can see this whole process is happening because of the CAMP only. So the CAMP is responsible for this all process. So that's why we are calling it as a CAMP response binding element binding protein. When the phosphate get attached on the CRIB, CRIB will go and attach with this CBP. CRIB binding protein means it is binding with the CRIB, so that's why we are calling it as a CRIB binding protein. And CRIB and CBP both will go and do transcription of DNA and make RNA. Okay, after formation of RNA will move into the cytosol region and it will do whatever the response cell should need to do. Okay, it could be any sensory response. Okay, here we are having another GPCR where we will cover about the PLC pathway. Okay, what is this? Let me try to explain you. You know very well, this is our plasma membrane, and on the plasma membrane 7 times GPCR receptor is crossing and it is made up of protein. Also it have N and C terminals. Okay, and our ligand only will attach on the E3 E4 center. When the ligand will come and attach on the N terminal side, C terminal will get activated and GEF will get activated. GIF will attach GTP, means it will provide one phosphate. So the alpha subunit will get a GTP, as you have already seen in the first pathway, right? Here, whenever the alpha subunit will get attached with the GTP, beta and gamma will stay here only, but our alpha will move towards the phospholipase PLC. We were covering about first GPs here. Okay, what we are covering about here, we are talking about the phosphorylase. Okay, phospholipase. Sorry. And what we adenylate cyclase we have seen in the first GPCR signaling. So the PLC means phospholipase is there, which is responsible for the breakdown of the PIP2. Okay, PIP2 molecule. So whenever the PIP2 molecule get breakdown, what it will form? It will form one molecule of DAG and IP3 molecule. This DAG is lipid soluble, so it will remain on the plasma membrane, but IP3 can move inside the cell. So IP3 will go towards the ER, means endoplasmic reticulum, and attach on the plasma membrane of ER and make a channel, so that the whole calcium, means some amount of the calcium, will release from the ER. After releasing of calcium, few calcium will attach with the calmodulin, and you can see the calcium and CAMP kinase. The 3CC will attach and make CCC complex. After that, it will do phosphorylation of any protein, so the response gonna happen. It could be any response, sensory response. And few of another calcium, you can see it will move and attach with PKC, protein kinase C, and the both will go towards the DAG. You can see here in the DAG, which is present on the plasma membrane, and PS also present beside the DAG. The DAG and PS, the both will responsible for the activation of our PKC. So the PKC will get activated and it will do protein phosphorylation or any response gonna happen in the cell. So this is what's happening inside our cell. Another thing we have to know about Vagra condition. Okay. In general, normal condition without Vagra, what is really happening, you have to understand. The CGMP present in the body, which will convert it into GMP. Okay. In the presence of phosphodiesterase, as we know, phosphodiesterase means nothing but the breakdown of the diester bond, right? The CGMP is converting into GMP in the presence of phosphodiesterase. Because of what will happen, the erection of the pineal will not happen. Okay. So what Vagra will do? Vagra will come in this condition and it will block the phosphodiesterase process. So the blocking of these phosphodiesterase means the CGMP will not form anymore. So the GMP will not form anymore, but the CGMP concentration will increase, because you can see very easily understand, we are not forming GMP with the help of CGMP. So CGMP remain as it is and GMP will not form here. Because of this, what will happen, the concentration of the CGMP will increase, so the signaling will start increasing in the pineal side. So because of that, what will happen, vasodilation will happen, and because of that, relaxation process gonna happen and pineal erection gonna happen. So whenever the person who's suffering with the pineal erection in especially male body, the doctor will suggest to take the Vagra. And we also must need to know there are two things are present in our vasodilation and vasocontraction. You need to know. This is vasocontraction and this is vasodilation. Okay. So when we talk about vasocontraction, the arteries and the bloodstream pathway, which is made up of blood veins, they are in the narrow condition. Okay, so the is called as a vasodilation, vasocontraction. Okay, but in the Vagra condition, the CGMP and signaling will increase the narrow condition of the vasocontraction into the dilation process, where the arteries will be get wider, so the blood can flow easily and signaling can be increased in this condition. Okay, you can see this is a narrow pipe of the blood which is traveling only few amount of the blood molecules, but here in the wider one where huge amount of the blood will travel, so the signaling will happen easily. So you can see also the relaxation of the muscle also present. Okay, what will happen here, the whole relaxation means completely in the relaxation position, so that relaxation is causing the pineal erection, so the solidify of the pineal will happen, and that all whatever the another thing you have to know very well. So this is what really happen in the Vagra condition. Okay, Vagra is blocking this all process. You can see the contraction and dilation process here in this image also. This is narrow condition which is known as vasocontraction, and this is vasodilation, wider one. Okay, now we will cover about the GPCR signaling in the artery system. Okay, here when we try to understand what really we are talking about, there are three layers are present in the arteries. Okay, you can see here the three layers are present which is making blood artery. One is tunica externa, another one is tunica media, and third one is tunica intima. Okay, these all layers are really important, you have to remember. Also you must need to know the narrow condition is this which is known as vasocontraction, and the wider one is known as a vasodilation. Okay, so what we are doing from this narrow condition to we are creating the dilation process, you can see here, and we are using NO nitric oxide for this whole process. And what we are doing, hydrophobic, these are the hydrophobic condition. Okay, there are two layers, one is tunica intima, another one is tunica media present here. Okay, this is tunica intima, this one is tunica media. You can see here also, this is our tunica intima and this is our tunica media. Okay, this one. So first we need to know the blood will travel through these veins. Okay, what will happen in that process, these blood streams also will carry some receptor GPCR receptor present on the tunica intima, where the ligand will come. Ligand is nothing but acetylcholine will come and attach here, then NO will form and give to the media, means tunica media. So here what will happen, dilation process will happen in the tunica media. Let's try to understand how this is happening. So see here, this is our tunica intima, where acetylcholine will come and attach on the GPCR. I don't need to say about the GPCR receptor how it looks. What will happen after attachment, GEF activation gonna happen. So the alpha will move towards the PLC, phospholipase, and PLC will break the PIP2 into DAG and IP3 molecule, you know very well. IP3 molecule will move towards the ER and attach with the plasma membrane and remove all the calcium from the ER by making channel. After calcium release, it will bind with the calmodulin, CAM kinase, and the CCC complex will form here. This CCC complex will go and activate the NO synthase by attaching of the phosphate. So NO synthase, after it will get activated, it will go towards the arginine amino acid and form citrulline and NO gas. Okay, after formation of NO gas, this NO gas will move towards the tunica media. Okay, we are coming on the tunica media here, where the guanyl cyclase present on the plasma membrane. On that NO gas, nitric oxide will come and attach here, and it will form CGMP. As we know, this is CGMP is our secondary messenger. This can be formed with the help of GTP. After formation of cGMP, this cGMP will activate our protein kinase G by active attachment of phosphate. So the PKG will get activated. This PKG is responsible for the protein activation. The protein will do the vasodilation. So the vasodilation, this wider condition can happen because of these all processes. And we also must need to know the acetylcholine can do three functions. One, it is doing the vasodilation process here in the arteries, and also it is responsible for the salivary secretion, and also it is responsible for the contraction of the muscles. So if you are taking some tablets for the dilation of the arteries, it will not only work on the dilation of the arteries, it can work on the salivary secretion as well as on the muscle contraction. So that's why people used to say that consumption of medicine not only affect the area where you want, but it can affect on other regions as well as. So the taking of tablet could be dangerous for health. I hope you understand it. Let's try to understand more GPCR signaling interesting things. Here we will see about the vision pathway where GPCR signaling is responsible. When we try to know about GPCR signaling, we have to know there are two cells are present. One is rod cells, which is responsible for the vision. Whatever you are seeing, that all are responsible for because of the rods. And another cells are present like cone cells, which are responsible for the filling of the color. You can see the eyes color, you can see this peach color and red color, black color. These all are responsible because of the cones. See, you can remember like a C for cone, C for color. Okay, rods are responsible for the vision. We will not cover about the colors, but we will not cover about the colors, but we will see about the vision pathway, how we are able to see. So for that we need to know about the rod cell. You can see the rod cell is present here. This is the structure of rod cell where one disc like structure is present. You can see this is our disc in which is present in the rod cell, and with the disc there is GPCR receptor also present. This GPCR receptor is known as rhodopsin. Okay. Also, the mitochondria is present in the cell and nucleus present, and remaining space is made up of cytoplasm. Okay, this is our rod cell. Now you need to know you already know about the rod structure. You have to see this is also how the signaling is happening. This is our rod cell. Okay, and here the disc is present, and this is our GPCR receptor. Okay, you can see here and compare so that you can easily remember. This is our complete cell, and here the disc is present. On this disc GPCR is present. Same like things are present here, GPCR receptor and cell and disc. Let's try to understand how this is happening. Let me zoom it. Okay. Now let's see. Whenever light will fall on the rhodopsin, means GPCR, what will happen? We know very well. There is alpha subunit will get activated, means GTP will get attached on it, and GIF activation happen here. Okay, this alpha will move towards the phosphodiesterase effector, and this alpha will activate phosphodiesterase and start making the GMP. Okay, with the help of cGMP. So the cGMP is making GMP, so the cGMP concentration will start decreasing in this condition. Okay, because of the decrease of this cGMP, here what will happen, negative things gonna happen in this condition. The cGMP gated channel will get closed. Also here the voltage gated calcium channels also will be closed in this condition. So we can say there is no more sodium will come inside, and no more cGMP is increasing, and instead of that the sodium concentration decreasing more, because the sodium is going outside the cell and potassium is coming inside the cell. Okay, sodium potassium channels are on in this condition. So what we can say here in this all scenario, The CGMP is decreasing as well as sodium is decreasing. So the hyperpolarization condition is happening and minus 90 volt gonna happen in the cell. And because of that, the glutamate will no more release from the cell of the rod. This glutamate is nothing but neurotransmitter. Because this is not releasing from the cell, so what will happen? Vision gonna happen. Okay, so not releasing of the glutamate is causing the vision in this condition. Are you getting my point? There is nothing easy lemon squeezy. You know very well this here till here the signaling process. Okay, CGMP is forming GMP, so CGMP concentration is decreasing and our CGMP gated and sodium channels are closed, so the sodium is not coming here and our calcium also not coming because of they are closed, but the sodium potassium channels are open, so the sodium is going outward and potassium is coming inward, but still the sodium concentration is decreasing, so the hyperpolarization is happening and minus 90 volt is happening, so the glutamate won't be able to release from the cell and we can see whatever we are able to see. This is what happening. But if I want to stop vision process, okay, I don't want to see anything, I kept you in the dark room. In this condition, what will happen? The light will not form, okay, fall on the GPCR rhodopsin. GPCR and rhodopsin both are same. Rhodopsin is our GPCR, GPCR is our rhodopsin. So when the light is falling, then only GPCR rhodopsin is activating, means photon is activating GPCR, alpha get activated. But for example, here in this condition, light is not falling on the GPCR, so the alpha will not attach here, so the GMP will not form, so the CGMP concentration remain high, so the sodium calcium also start increasing in this condition and our hyperpolarization condition will not happen. So here depolarization condition happen here in this condition. Because of that, glutamate will make vessels here and it will release through the plasma membrane cells, okay, and also releasing can, okay, release, after releasing what will happen? No vision gonna happen. So this is what happen when we do not want to create the vision, this glutamate is releasing. But if we want to create the vision, then the glutamate is not releasing in this condition. How interesting it is, right? Let's try to understand in this point of view, so you will get easy understanding. So on the GPCR rhodopsin, light is falling here, so the vision is happening here because of not releasing of what? Glutamate. The light also called as a photon. Okay. Let's try to understand what is rhodopsin. And our rod and cone cells are present here in the retina, peripheral retina region. You must need to know. And rhodopsin is made up of two things. One is protein opsin and chromophore. Chromophore is derived from the vitamin A. That's why doctor used to suggest always take vitamin A for good vision. And this chromophore is having 11 cis retinol. Okay, so the rhodopsin is made up of two things. One this and another one this. Whenever light is not present, okay, the light is not present, opsin and 11 cis retinol is present in the dark condition and they are in the inactive condition. But whenever the light will come and fall on this condition on the rhodopsin GPCR, the opsin will remain same, but our this 11 cis will become 11 trans. So 11 cis will change the structure and form our 11 trans. Here the trans should need to write. The 11 trans retinol will form and it will create the vision here. So this 11 trans is always unstable in the condition. After a time, whenever you will again go into the dark or you will close your eyes, opsin will remain same all the time constantly, but this 11 trans retinol will convert as to the 11 cis retinol in the dark condition, so that rhodopsin will form. Okay, so this is what happening whenever we are creating vision. We are having these all processes are ongoing. So option always same in every condition, but 11 says is always in the dark condition, means you can see think like you whenever you are seeing, means you are seeing right now, so your trance is activated, it is unstable and you are able to see. If you close your eyes, your cis will get activated and the inactivation of the rhodopsin is happening. That's it. Again you will close open, then 11 trans will become cis and cis become trans. This is what really happening in the retinol region. We will cover about the cholera toxin. This is also comes under the GPCR signaling. What really happened, the cholera toxin, the diarrhea condition is formed by the cholera toxin. This toxin is called as a vibrio cholera. This is nothing but enterotoxin. What is enterotoxin? The enterotoxin is nothing but the intestinal, anything which is causing any effect in the intestine called as a enterotoxin. The enterotoxin is made up of two units, okay, two subunits. Just to wait a second. Let me clear my screen slightly, it is stuck in here. Okay, let's try to understand. So the cholera toxin is a toxin which is responsible for the diarrhea in human being. This cholera toxin is called as a vibrio cholera. Okay, it is causing enterotoxin in human being, means intestinal problem. It has two subunits. One is monomer and another one is pentamer. Monomer is present in the center region, which is known as A subunit, and pentamer is known as B subunit, which is present outer region. It is around 5 subunits are present. That's why we are calling it as a 5. This 5 and this 1 is known as hexamer. Okay, let's try to understand how this is happened. In general, in normal condition, even though right now if your body is in the normal condition without any diarrhea, what is happening? See, first you need to know normal condition, later we will see the diarrhea condition. So the normal condition, when you are not suffering with diarrhea, so our GPCR receptor will be get a bond with some ligand. This ligand could be anything, it could be hormone or adrenaline, any hormone. After binding of a ligand on the GPCR, as you know, C terminal means GEF activation gonna happen here in this condition, and this all process is happening in the intestinal cell. You can see this is our intestinal cell and this is extra side of the lumen of the intestine. From here the body waste material will gonna travel. Okay, so inside the cell the GEF get activated and our alpha subunit get attached with the GTP and it will move towards the adenylate cyclase, or you can call as a adenylyl cyclase. Adenylate cyclase get activated because of the alpha subunit get attached with the GTP here. So the GTP and GTP will activate adenylate cyclase, and here the CAMP will start formation with the help of what? ATP. This CAMP will activate pKa, protein kinase A. So the pKa will go towards the chloride and sodium water channels. What it will do? It will remove chloride from the cell, it will remove water from the cell, and it will take inside the sodium. So this is what in the normal in general condition is happening. But whenever the cholera toxin will enter in our body, what will happen? Let's try to understand. Here how the cholera toxin will enter. There is a ganglioside receptor is present on the plasma membrane. This ganglioside receptor will get attached with the cholera toxin, but the whole hexamer will not enter. Only monomer condition of A subunit will enter inside the cell. with the help of what? Endocytosis. Okay, A subunit will enter in this cell with the help of endocytosis. Okay, after entering of the A subunit, the 5 pentamer subunits remain outside the cell, outside the body. But the A subunit will do ADP ribosylation of the adenylate cyclase. Means what it will do? It will attach ADP on this alpha subunit. So here you can see the ADP get attached on the alpha subunit. So the ATP is continuously form CGMP. So the CGMP concentration increase in the cell. Because of that, pKa concentration also increase, and because of this they will attach, and constantly it will remove the water and chloride from the body. So normal condition where the alpha is activating adenylate cyclase, but this A subunit is constantly activating our adenylate cyclase, as well as constantly activation of the channels are happening. So the electrolytes of the body will be decreased in this condition. Also, it will cause lots of fatigue and dehydration in the body. So that's why the people will suggest to take the ORS. Okay, in this condition your body need ORS electrolytes. So this is what really happening inside the body. Another thing we will cover about the glycogen, which is also connected with the GPCR signaling. Here in this condition we must need to know there are two hormones are present. One is insulin, another one is glucagon. Insulin and glucagon, they both are released from the pancreas, but the situation of releasing is completely different here in this condition. The insulin will release whenever you intake of lots of sugar, then insulin will release, and this insulin is responsible for the making of glucose into the glycogen. You can see here also. Okay, so insulin is responsible for the formation of glycogen with the help of whatever the intake of the glucose you have taken. But glucagon will release when the body is not having enough amount of the glucose in it. So we can say whenever you are fasting or you are not having food because of the anger issue, the glycogen which already stored in your body will break down and form glucose. Okay, don't get confused here in these terms. Glucagon is a hormone, glucose is our energy molecule, and glycogen is our glucose attached molecule. Okay, we can't store glucose separately, so we will form glycogen. Bunch of glucose are present in the glycogen molecule, they can be stored easily. Okay, this is known as a glycogen. So this is what happening. So we can also say, I hope you are getting, we can also say this glucagon can release when the blood sugar is decreased. So the blood sugar can increase with the help of the formation of glucose. And we can see the insulin is released when the blood sugar is high. So it can decrease the blood sugar level. Okay. And also we need to know there are some synthase present, glycogen synthase is present. Okay, in insulin, which is in active condition, and glycogen synthase is phosphorylated in this glucagon formation, where the phosphorylation will become inactive condition. Okay, how? Let's try to understand. As you know, we know about GPCR receptor. I don't want to say again and again. The glucagon will come and attach on the receptor. Okay, and you must need to know one thing else also. For example, insulin release, it will go in our bloodstream and it will give response to the our liver and muscles. So that what will happen, they will store and form glycogen and store in the body. Okay, so this is, it could be our liver cell or it could be our muscle cell. Okay, where the glucagon will come whenever you are not having food, and glucagon will attach here on the receptor. So the C terminal get activation and alpha get the GTP on it. It will move toward the adenylate cyclase and form CAMP with the help of ATP. The CAMP will start formation of pKa protein kinase A. The till now you know very well, but from here you need to understand there will be two process can happen. These pKa could inhibit glycogen synthesis, or it could do stimulate the glycogen synthesis. When we are doing these two different pathway, whenever your muscle cell or your liver cells are completely filled with space, okay, for example this is the space of the muscle cell and you are storing some glycogen molecules, okay, glycogen, everywhere is glycogen present and you don't have space. At that time what you need to do, you need to inhibit the process of synthesizing of the glycogen synthesis. So what you will do, you will phosphorylate on the glycogen synthase, so it will become inactive and no glycogen will synthesis here. But you have lots of space, okay, and you want to release good amount of the glucose outside the cell. At that time what will happen, glycogen phosphorylase kinase will get converted into the glycogen phosphorylase kinase phosphate, okay. The both names are same here. The glycogen phosphorylase kinase is get activated by the phosphorylation. After glycogen phosphorylase kinase get activated, it will activate glycogen phosphorylase. You can see the names are completely same. The kinase is present here in this condition, there is no kinase is written. So the glycogen phosphorylase get activated with the help of glycogen phosphorylase kinase and it will get phosphorylated, means glycogen phosphorylase get phosphate. After phosphorylation, what the glycogen phosphorylase do, it will do the glycogen breakdown and glucose 1-phosphate gonna form here. From here the blood will start increasing the glucose amount, so the energy can be used from the body which already stored, okay. Then you must need to know another thing. For example, in our body the glucagon is not attached. At that time what gonna happen, there is no CAMP gonna form, there is no pKa form. So in this all condition phosphatase will come. Already I think you have remembered, phosphatase is present, phosphorylase is present. Phosphatase means it will remove the phosphate from any protein. So here in this condition you can see glycogen phosphorylase kinase get removed its phosphate, so it will be get inactive, okay. Here in this condition glycogen phosphorylase will get removed phosphate. Here the kinase is present and there is no kinase, so this one will remove the phosphate, so it will be get inactive, okay, inactive. Then we are having glycogen synthase which also get activated, which also get attached with the phosphate, but it is in the inactive form. So that's why the removal of phosphate will give the activation scenario here. So then the glycogen synthesis is gonna happen, okay. I think you will definitely get confused here, okay. Let me try to explain you in this condition. This whole chart I am explaining here, okay, so that you can understand it clearly. In first condition we were seeing about glucagon attaching, so that gets activated, CAMP form, pKa could inhibit or stimulate, so the glycogen phosphorylase kinase get phosphate and phosphate here also attached, so the glucose is forming. This is what happening. But we want to know another thing also. For example, there is no glucagon is attached on the GPCR receptor, so there will be no GAF activation is happening, so the CAMP will not form, so the pKa also not form. In this condition phosphatase will activate, okay, phosphatase, sorry I have written wrong, phosphatase gonna be activated, okay. This phosphatase has the ability to remove phosphate, right. What it will do, it will remove this phosphate, means glycogen phosphorylase kinase phosphate gonna remove, so it will become inactive, so there is no phosphate. It phosphatase also removes the phosphate of these glycogen phosphorylase. So this is also go in inactive. Okay, inactive condition, inactive, right? But here the glycogen synthase is inactivated when the phosphate is attached. When you remove this phosphate with the help of phosphatase, what will happen? This inactivation become activate, or glycogen synthesis will happen. Okay, so this is what happened when the phosphatase will come. I hope you understand. If you see this slide 2 to 3 times, you will understand. Otherwise, everything will fly out of from your brain. Last GPCR receptor concept, desensitization. I think this one is the last. Yeah, this one is our last topic about the GPCR. Desensitization. Here what is happening, let me try to explain you. Why we are doing desensitization? If receptors are too high on the plasma membrane, for example this is our cell and we are having lots of receptor on the plasma membrane, so what will happen? For a particular signaling we don't need these many receptor, so we need to remove them. Okay, few of them should need to remove. This is one situation. Okay, other situation where we are having normal amount of the receptor, but the signaling is going too much, means ligands are binding and attaching and signaling is working too much. So in that condition what we need to do? We need to do desensitization process. Let's try to understand how it is working. So we are having GPCR receptor on the plasma membrane. It could be any plasma membrane GPCR, wherever it could. Whenever ligand will come and attach, alpha get activated and alpha will go towards the adenylate cyclase, and adenylate cyclase will get activated, form CAMP and pKa form. Okay, and it will go in the nucleus and it will activate our CREB. CREB get phosphorylated, so the CREB will bind with the CBP. Till now you know very well. Okay, these CREB and CBP both will go and activate the GRK gene. So the GRK gene will form GRK mRNA. So GRK protein gonna form here. This GRK protein called as a G protein coupled receptor kinase. You know very well kinase means phosphorylation. Here what will happen? This GRK protein will use ATP and it will do feedback inhibition. So the receptor should need to stop. Okay, this ATP will used here and it will go and attach on the GPCR receptor and it will do the phosphorylation of the GPCR receptor here in this condition. After phosphorylation, arrestin protein will see and understand something is going fishy. So the arrestin protein will come and attach on the GPCR receptor, and clathrin also come and attach. The clathrin function is nothing but it will make vessel, and so the endocytosis is gonna happen and the vessel gonna be formed here in this condition. The GPCR will take inside the cell and it will send towards the early endosome. From here the GPCR could be sent towards the lysosome or it could do the recycle process. As I said to you, if there are so many receptors, then we will send these GPCR into the lysosome, so degradation gonna happen in this condition. But we are having only few receptors because the formation of receptor take too much work. So in that condition, if the process is stimulation is too much, so in that condition we need to send back our receptor, recycling will happen, and again the GPCR receptor will attach here on the plasma membrane. So as we are taking inside and we are putting back, in that all processes what really happening, in between this all time this stimulation process gonna decrease in the cell, so the signaling will not work too much, extra stimulation not gonna happen too much, also bad for everything. So same thing like that. Okay, this is going to work as a feedback inhibition. Why? Because we are saying, if you think like if you are having food and someone is came to you and serving you food, the plate get fulfilled, then you have to say stop it. You need to, okay, you are putting some food, giving some food does not mean you have to do give that until the plate will overflow, right? you need to give feedback just to stop. It is enough for me. So same like that, the stimulation and desensitization process is happening. So the GPCR will stop here and the stimulation process need to shut down for a few minutes. For this whole process also it will take long time. So in between that the GPCR is completely inactive condition. So this is about desensitization. Next video we will cover about the RTK pathway. Okay, till now if you understand each and everything and you felt it's an amazing class, then please give me some comments and give thumbs up, and please subscribe to this channel so I'll get lots of motivation to make more videos, or else I feel this is not worthy for anyone, so that's why my motivation will drop out. So I feel you enjoyed it and you understand each and every concept. Even though if you have any queries, anything, you can ask me in the comment section. Thanks for watching.